Hi, this is the Cell Biology Reverse the Classroom video. Now this is then just kind of a bit of an overview, right, of everything that we've kind of got. We've got cell structure, we've then got eukaryotic and prokaryotic, we go to specialized cells, we've got a required practical in here using the microscope, we've got stem cells, we've then got binary fission, chromosomes, mitosis, diffusion, exchange surfaces, osmosis, osmosis required practical, active transport. That is then kind of an overview of the whole unit. Now, kind of one of the most basic things of everything, right, and it feeds into a lot of different areas, is the structure of an animal and a plant cell. Right, so just looking at the plant cell first. So the plant cell has got a chloroplast, it's got a vacuole, it's got a cell wall, it's got a cell membrane, it's got a nucleus, it's got cytoplasm, it's got mitochondria, and it's got ribosomes. What you can do is you can notice that the plant cell has got chloroplasts, it's got vacuole, and it's got cell wall that are different to the animal cell. Because what an animal cell has, it has ribosomes, mitochondria, cytoplasm, nucleus, and cell wall, cell membrane rather. Right, so quite often what you might do in an exam is you might get asked what are the extra three things that a plant cell has got, and that would be chloroplast, vacuole, and cell wall. Now I think one of the best ways of remembering it is just do something like this, where what you can do is you can just name the numbers, all right, as you actually go around. So we've got number 1 is the chloroplast, number 8 is the vacuole, number 7 is the cell wall, number 6 is the cell membrane, number 5 nucleus, number 4 cytoplasm, number 3 number 3 mitochondria, and number 2 ribosome. Now you also need to know the functions of each part. So what the cell wall does, right, which is only in the plant cell, it supports the cell and keeps its shape. The nucleus, it contains DNA and controls the activity of the cell. Mitochondria is respiration. This is then where energy is created or released. Vacuole is full of cell sap and maintains the shape of the cell. A cell membrane controls the movement of substances in and out. Ribosomes are proteins. I don't like made, I like synthesized here. So it's protein synthesis. Cytoplasm, this is where many reactions take place, and again what I'll do is into that one I would then write chemical, right, it's where chemical reactions take place. And chloroplast is light energy absorbed, and this process is photosynthesis. Now another structure that you also need to know is your bacteria. Okay, now a lot of the things in a bacteria are similar to the plant and the animal cells. Okay, so if we go for cytoplasm, okay, it's got exactly the same function, it's where chemical reactions happen. We've got cell membrane, which allows, controls what substances go in and out. We've got a cell wall, which maintains the structure. And then we've got others that are then slightly different, where we've got plasmids and we've got genetic material. Now what they are is they're the genetic material, the DNA, but it's not inside a nucleus. Then there's also two others. We've got a slime capsule and we've got flagella, right? They are sometimes present, where the other 5 are always present. So the slime capsule, I would kind of envisage that as being kind of like a sticky layer around the outside. So what the bacteria can do, it can stick to something. And then the flagella is then there for movement. Now, what we just talked about, we've talked about animal and plant cells, and we've talked about bacteria. So animal and plant cells are your eukaryotes. And I will kind of think of it as you as a human are a eukaryote. Right? So you are eukaryotes. And what a eukaryote is, the main thing about it is it contains a nucleus. But it also has other organelles that have a plasma membrane around the outside of them. So for example, where we've talked about mitochondria before, can't really draw a mitochondria, right? What it's got kind of like a bag around the outside, which is your plasma membrane. So it's an organelle that is bound by a membrane. It's like in a bag. A prokaryote is your bacteria, right? And so what a bacteria does, it does not have a nucleus. And it also doesn't have membrane-bound organelles like your mitochondria. So mitochondria, right? And what a bacteria does, it doesn't have them because it's a membrane-bound organelle. But the key to it is eukaryotes have a nucleus, prokaryotes don't have a nucleus. Okay, that's the basics of it. Now, onto microscopes. Now, what we've got here is a very basic microscope, and we've only got 5 kind of little labels on it. So we've got the eyepiece lens, which is obviously what you look down. We've got the focusing wheel, which sometimes what you'll have is you'll have a coarse focus and a fine focus. Coarse focus is to get it approximately right. A fine focus is then to get it accurately right. We've got the objective lenses. Usually, again, there's 3 of them, and what you do is you spin them on this kind of nose part here, and it could be times 10, times 4, times 40. Okay, it could vary depending on what microscope you've got. You've then got the stage, which is then where you put the microscope slide, and you've got a light source. Sometimes what they'll have is they'll have a mirror here, and what you'll have is you'll have light shining here from a bulb, hits the mirror and goes straight up into the objective lens. Now what there's a required practical. Now the best way of doing this is go to my other video that then goes through the required practical itself in a lot more detail. But in a nutshell, what you've got is you've got 2 different parts to it. You've got how to prepare a slide, and it's usually an onion cell, and you've got how to use the microscope. Now that's pretty unusual to actually be asked about how to use the microscope, but preparing a microscope slide is one that comes up quite regularly. But like I said before, go to my required practical on microscopes and see it in more detail. Now, comparing light and electron microscopes. Light microscopes, they use light rays. Electron microscopes use electron beams. Okay, now these next 4 parts here are the best ways of comparing. So magnification. I would always say a light microscope has low magnification. An electron microscope has high magnification. Resolution. I'll talk about the definition of resolution in a second. Resolution is low in a light microscope, and it's very high in an electron microscope. Size of the microscope. So they're small and they're large. Cost, £100 for a school one, 100,000 to £1 million for an electron. So they are cheap and they are expensive. Okay, then, like I said before, a couple of little definitions. Resolution is the ability to distinguish between 2 different points in a specimen. It's a bit like your TV screens, right? If you've got 2 TVs, that one there is 4K, that one is the old 1080p, right? 4K means it's got a lot better resolution, which means there's more pixels per centimeter squared than the 1080p one. So the resolution, the clearness, the ability to distinguish between 2 different points in a specimen or on a picture, the 4K one is better. Magnification is how much bigger an object appears compared to the original object. So a light microscope is quite low. An electron microscope has a very high magnification. Now, conversions. Now, these questions come up a lot. So what I've got on here is I've got a little diagram that kind of converts between 5 of the main ones. Now, there is another one that could come in here often, which is centimeter. Okay. Now, the reason why I've done these 5 is because there's basically 1,000 times or 1,000 divide between each one. So if you want to go from meter to millimeter, you times by 1,000. right? Because if you think about it, you've got a meter stick, so that's 1 meter. How many millimeters are in 1 meter? There's 1,000 millimeters in 1 meter. To go from millimeter to micrometer, look at the actual symbols. The units is 1,000 again. Then a nanometer, which is nm. A picometer is then pm, which is then 1,000 times again. These are then the standard form based on a meter. To convert back in the other direction, what we've then got is a picometer into a nanometer, etc., etc. What you really need to do is you really need to remember these 5 things in order with the units, so then you can convert between any of them. If you're ever asked about microscopes in your exam or in any form, what there will be is there will be a question to do with calculating image size or magnification. And the way I remember this, and it's the only one of these triangles that I use regularly, is I remember it as the AIM equation. Okay, it's the AIM equation. Actual, image, and magnification. So what you can do is, if you need to work out the image size, you put your hand over the eye. Okay, I'm just going to scribble it out, even though that's a very bad way of doing it. And it's actual times magnification. Now, specialized cells. What we've got is we've got 6 that we need to go through. 3 animal and 3 plant. Starting off with a muscle. What we've got is we've got 3 different types of muscle cells. We've got cardiac, which is in the heart. We've got skeletal, which is all connected to your skeleton and your bones. And we've got smooth muscle, which is mainly your digestive system. Skeletal are voluntary, which means we can control them. Smooth muscle are involuntary, which means we can't control them, as is the cardiac. Now the specialism is, it's really only one main one, is the fact that a muscle can contract. Okay, so what we've got here is we've got a relaxed muscle. Then what the muscle cells can do is they can contract. And that's the only thing they can't do. They can't push, they can't do any of those different things. All a muscle cell can do is contract. So that is its specialism. A nerve. Okay, now this is a relatively complicated picture of a nerve, really, right? But what you've got here is you've got the nerve endings, right? The nerve endings could be in your skin. And at this end here, the dendrites is kind of where it all sort of gets distributed around the body. So what you'll have is you'll sense something here. It goes down this orange part here, which is the axon. Then what you've got around the outside, you kind of got insulation, so you don't lose your actual nerve impulse. Then what it does, it gets distributed via the dendrites. Now the main ways in which it's actually specialized is the fact that it's long. It has the ability to conduct electricity. Okay, they're the kind of the 2 main specialisms. The sperm. Okay, now sperm's quite incredible, really, right? It's got numerous different specialisms. It has the acrosome, right? And now what the acrosome is, it's like a little bag of enzymes on the head of the sperm. So when the sperm actually swims into the egg itself, the enzymes burst and it helps the sperm get inside the egg. Inside the nucleus, it's got half the number of chromosomes. Okay, so for humans, it'll have 23 chromosomes, and the other 23 are in the egg. In the neck, what it's got a large number of mitochondria. Mitochondria. Right, that is then because of the energy that's then required. And it also has a flagellum. You probably get away with calling it a tail. That is then for movement. Okay, so the sperm has got quite a number of different specialisms. Root hair. Again, this is quite a complicated picture of a root hair. So what you've got on a root hair is, right, if I draw a very, very spectacular tree, Okay, that is obviously a tree. Right, I'll put some leaves on it, even though they now look like a flower. Right, what you've got is you've got your roots underneath the ground. On each one of those little roots there, what you've got is you've got loads of root hairs. Okay, and that's pretty much what that's supposed to be. And the whole specialism with a root hair is to increase the surface area. Okay. Now, the next two are xylem and phloem. Okay, so we've got xylem and we've got phloem. Now, the specialisms for xylem is the fact that what xylem can do is it carries water and minerals from the soil. It has no end walls between the cells, so what it means is these here, because they are dead, it only transports stuff one way, which is up, right? And then it says here outer cells are not living. Okay, and this process is transpiration. The other one is phloem, which is a little bit more complicated. What it'll do, it'll transport organic molecules, often glucose, made in photosynthesis. At the end of each cell, it has things called sieve plates, or they can be called end walls. And what they are is they are the part that actually decides where things go. There's two-way movement, so it can go up and down. So if you've got your tree, right, there's your tree again. And what you can do is if you make some glucose there, what it can do, it can go to another leaf over here, or it could go down to the roots down here. They are living, the actual phloem cells. And the process that occurs within phloem is translocation. Okay, now onto DNA. Now, I have seen this question come up a few times, which is where it tells you to organize sort of the DNA smallest to largest, or things to do with it. Right, so what you can do is, there's your cell, which is number one. Okay, that's the biggest. Inside the cell, what you then got is you've got the nucleus, which is the next biggest. Inside the nucleus, you've got your chromosomes. Okay, so chromosomes, they're usually drawn as an X shape, right? Even though normally in a cell they're just kind of like one strand. DNA is then the chemical that is actually made up. So the whole sort of double helix, I'll just write down there, double helix is your DNA. And then you've got genes, so there and there is a gene. And that is like one part of the DNA. So what you might do is you might get asked to talk about it from the organization within a cell of DNA, largest to smallest. Make sure you do it that way. But they do try and catch you out sometimes by doing smallest to largest. So just be very careful. Now, the cell cycle. Again, this is kind of a bit of a question. Often what you might do is you might get a pie chart that looks like that. You might get asked to name any of the individual parts of it, or what happens in each individual part. So the first one is interphase, right? And what that is growth and replication, right? So it's basically everything is doubling, because the whole point in this cell cycle is to create two cells. So starting with one cell, ending up with two cells. So in interphase, everything doubles. So things like the mitochondria will double. The ribosomes will double. Okay, the actual DNA itself will double. Okay, that is interphase. Right, number two, what we've then got is we've got mitosis. Right, and on the next slide, I'll go through the process of mitosis a little bit more. Right, this is then when the actual nucleus divides. And the final part of it is cytokinesis, which is where the entire cell then splits. So what it has one cell like that that's kind of doubled up in size, and what it kind of does, it then breaks up into two other cells, and that's cytokinesis. Now, mitosis itself. Now, what they'll normally do is they'll normally have one or two or four pieces of DNA in there. So what you can do is you can see we've got a red one there, and we've got a blue one there. They're two different chromosomes. First thing that happens is they double in size. So what you got is you've got a chromosome like that, and what you have is you have a thing in the middle, a centromere, and it kind of grows different parts like that. It's not quite as simple as that, and they're connected in the middle there like that. And the top layer and the bottom layer are then identical. So what you do is you produce two X-shaped chromosomes. Then what they do is they line up. Okay, I'll often sort of draw it where I actually draw them like that, where they literally line up down the middle. Then what they do is they separate, and what they do is they split on the actual centromere itself. So half goes that way, half goes that way. And then what you do is you make two identical diploid cells, right? And what they are is they are then daughter cells. And the one at the very start was then the parent cell. Now, because opinions, and there might be sort of ethics questions coming in, we're going to be talking about stem cells in a minute. So I'll just talk about a little bit from the Catholic Church. Roman Catholics believe the use of any embryos in research is morally wrong. No objective, even though noble itself, such as a foreseeable advantage to science, to other human beings or society, can in any way justify experimentation on living human embryos or fetuses, whether viable or not, either inside or outside of the mother's womb. So really that's saying is there is no experimentation allowed on embryos or fetuses at all, even though it could make sort of scientific research improved and new drugs could be made. Right, from a Catholic point, that just cannot happen. Now, the three different types of stem cells. We've got human, we've got human embryonic, we've got adult, and we've got meristems. So firstly, the two kind of ones from humans. Human embryonic stem cell, right, are very, very, very early, right? What they can be is, it says there, 5-day-old. So a sperm fertilizes an egg, and within 5 days what you've got is stem cells. And the fantastic thing about those stem cells is they are ready to be converted into anything. So what they can be is embryonic stem cells can be cloned, and they can be turned into most individual different types of cells. So there's a lot of research that's gone into human embryonic stem cells, where if you can inject them into a certain place on somebody's body, maybe it could be replicate or rebuild the part of the body that was there. The second type is the ones that we all have. All right, and what they are is they are adult bone marrow stem cells, or sometimes just adult stem cells. And what they can do is they can form many different types. All right, but they are limited. And the final type is meristems, right? I always call them the happy stem cells, right? And what they are is they are then plant stem cells. And what they can do is they can differentiate into any plant cell type throughout the life of a plant. Okay, so if you break a branch off a tree, what happens is stem cells will go to where that branch was broken, and new growth then could appear. Okay, diffusion. Diffusion happens in liquids, which is here, and it happens in gases, that is here. Now, the definition, right, is dead important that you understand it, and it's dead important that you remember it. So diffusion is the spreading out of particles of any substance in solution or a gas, resulting in the movement from an area of higher concentration to an area of low concentration without using energy. Right, and I'll also add into that one down a concentration gradient. Okay, so what that means is it begins to go from a high concentration to a low concentration. Right, so if you imagine there's a room, okay, there's a room, somebody walks in here with perfume on. Diffusion is the movement of the particles from a high concentration, which is here, to a low concentration, which is over here, down a concentration gradient without using energy. And eventually what will happen is all the particles will then spread out. Now, there are a number of factors that affect diffusion. If it's warmer, diffusion happens quicker because the particles are moving faster. Surface area, if there's a large surface area, diffusion will happen quicker. If there is a bigger difference between the concentrations, so higher gradient, diffusion will be faster. Size of the particles, if a particle is smaller, it will diffuse faster. And also diffusion can happen in all three of the different mediums, solid, liquid and a gas. It happens slowest in a solid, then a liquid and then a gas. I always think about it with blue tack. Right? If you think about blue tack, right, is that how you spell blue tack? Has it got a K on the end? I'm not sure. Right, that you stick blue tack on your wall, eventually what happens is the blue tack will kind of go into the actual paintwork. Right? And I guess that is kind of diffusion. Osmosis. Right, now osmosis is pretty much the same as diffusion, except it uses water. So I'm going to do this in blue, water. Now, so osmosis is the diffusion of water from a dilute solution to a concentrated solution. Right, so what that means is a dilute solution means there's high levels of water to low levels of water through a partially permeable membrane, or sometimes it can be a semi-permeable membrane without using energy. And again, it is down a concentration gradient. Okay, so this diagram shows it here. So what we've got here is we've got water and we've got a cell. And because there's a high concentration of water on the outside, osmosis is the movement of high concentration of water here to a lower concentration of water here across a partially permeable membrane. Now, sucrose can't go in the opposite direction because it's too big to go through the actual holes in the membrane itself. Now, there is an osmosis, required practical, and literally all I'm going to do on here is, this is like a slide that kind of goes through all the information. Best thing to do is to go through to the required practical osmosis video, and what you can do is then you can see all the bits of information that are is much better explained. Now, we've got diffusion, we've got osmosis. Now what we've got is we've got active transport. Okay, active kind of gives it away a little bit. So active transport is the movement of substances from a more dilute to a more concentrated. So that is against a concentration gradient across a semi-permeable membrane, but it does use energy. So this time what we've got is outside the cell we've got less, inside the cell we've got more. An active transport is where it goes against the concentration gradient. Okay, but the key to it is the fact that you're using up energy. And the last little thing then is exchange surfaces. Now, exchange surfaces are where there is transport, whether it's diffusion or whether it's active transport, across a surface. Now, if I just talk about the small intestine first. So what we've got is, that's kind of how you imagine your small intestine. But on the outside of all the small intestine, what there is, there's loads of kind of looks a bit like hairs. Right? And what those hairs are villi. And what they do is to help the exchange surfaces, help the movement, is the villi increase the surface area. So increase, I'm just going to put SA. Okay, so that's what the villi does. The villi also have a good blood supply. Okay, so the good blood supply is to maintain the concentration gradient. And they also have a thin membrane. Okay, so they've got a thin membrane because what that does then is that gives a short diffusion distance. So there isn't very far for all these things to go. Now, quite interestingly, we've got the small intestine. If I then start talking about the lungs, what the lungs have is they have alveoli, which have a good surface area, increased surface area. Alveoli have a good blood supply to maintain the concentration gradient, maintain it, and they also have thin membranes for a short diffusion distance. So what you can see is alveoli have exactly the same three reasons. Gills of a fish. Gills of a fish increase the surface area. They have a good blood supply to maintain the concentration gradient and thin membranes for a short diffusion distance. Right, so gills of a fish have got exactly the same thing. So if ever you see exchange surfaces on any question ever, they are your three answers. Increase the surface area, they've got a good blood supply to maintain the concentration gradient, and they've got thin membranes to allow a short diffusion distance. Now there are exchange surfaces in plants as well. We've got the roots, which has then got root hairs to increase the surface area, and we've got leaves. Leaves have a large surface area, thin for a diffusion or short diffusion pathway, stomata on the lower surface to let oxygen and carbon dioxide in and out. Okay, now answer these 5 questions right into your exercise book. Write out the question first and then give your answers following on from the written out question. Number 1 is, what are the 5 factors affecting diffusion? Question number 2, what are the 3 main types of stem cells? Question number 3, what is interphase? Question number 4, describe how a sperm is specialized. And number 5, compare a light and electron microscope, and I want 4 different comparisons for that.